In this video, I will be explaining the transfer of water and sugar to the phloem by translocation, indicated by water in white and sugars in black. Sieve tube members are the tubular-shaped cells that are one component of the makeup of the phloem, necessary for sugar and water transport. The second component of the phloem is the companion cell, which is the life support of the sieve tube member cell and has all the necessary organelles to keep the sieve tube member alive. Without this, the phloem is non-functional. The sieve tube plates are perforations at the end of each cell that allow and regulate bidirectional movement of sucrose to the adjacent sieve tube member cells. Contact between the companion cells and the sieve tube cells is mainly through plasmodesmata, which serve as microscopic channels which cross the cell walls. Sugar is first produced by photosynthesizing tissues in the palisade mesophyll, which act as the source. In the source, sucrose molecules are polymerized by the joining of a glucose and a fructose, forming a glycosidic bond between the two sugar molecules. Then sucrose is actively loaded, using ATP, and enters the sieve tube members by companion cells. When sucrose is actively loaded using ATP, this is referred to as phloem loading, which is the process of sucrose entering and leaving through the cell walls by active transport. In this case, the active form is apoplastic movement, which works similar to the ion exchange in xylem. However, in the phloem, the symporter pumps out protons and uses ATP to pump out hydrogen ions and then pairs with sucrose to move down the sucrose proton cotransporter into the sieve tube member cell. This is the alternate of symplastic movement through the plasmodesmata, which does not require energy due to the conjoined cells. Back to translocation, the movement of sucrose into the phloem causes the concentration of solute to build up in the sieve tubes, which creates a high concentration of sucrose. This high concentration of sucrose allows for the transfer of water through osmosis, where water enters a selectively permeable membrane from a low solute concentration to a high solute concentration from neighboring xylem vessels. As water is not able to be compressed and the sieve tube member has a rigid cell wall, the flow of water creates an abundance of internal pressure. This internal pressure of liquid is known as hydrostatic pressure. Therefore, hydrostatic pressure drives the mass movement of water and carbohydrates down from the source to the sink. At the sink, companion cells actively unload the sucrose from the sieve tube through the process of offloading, again using ATP and membrane-bound proteins to alter the hydrogen ion and sucrose gradient. Some of the carbohydrates are converted into starch and stored, while others are used for rest-firing cells. In response to the sugars leaving the sieve tube member decreasing the solute concentration, osmosis again moves the water to the neighboring xylem from low concentration to highest concentration. The loss of water from the sieve tube leads to a drop in hydrostatic pressure. This allows transport along hydrostatic pressure gradients in sieve tube members. As phloem sap flows from source to sink, it's transported from a region of high hydrostatic pressure to one of low hydrostatic pressure, translocating the energy and nutrients throughout the plant. In other words, translocation is the movement of sugars either up or down the phloem from source to sink. This process repeats as photosynthesis, polymerization, osmosis, and the transfer of sucrose continues.